out on for leave and she'll be out until May. I'm gonna, for the next two meetings, this one and one coming up in 2 weeks, where Jerry Zegers is going to lead the discussion. I'll help to coordinate. But do we have any announcements that folks would like to put out today? And annual reporting is ongoing. I think the process is coming together on that. And so on. So that's a major activity. I don't know if anybody else has other key activities to put on the radar, other than now that we're into March, we're starting to move back into some of our index site sampling and so on here coming up pretty soon. I think Sean is helping to serve as a point person for a lot of that, of course, especially also while Karen is away. And so those are the things that I know of on my radar. And there's also been some planning with folks starting to talk about the upcoming all hands meeting in May in terms of schedules and so on. So there'll hopefully be some news around plans for that before too long. And so with that, and if nobody has any items they'd like to chime in, humble brags or the like, I'll turn it over to Jeremy, Dave, Kate, and others who put themselves down as leading some of our discussion around synthesis opportunities, which of course is a big piece of where we need to be heading in remaining time on Maine eDNA. So Jeremy, Dave, Kate, please take it away.
Like, guess I'm gonna kick it off here. And so, yes, we want to get to synthesis discussions. We're still sort of focused on the index sites, and that's a big part of the synthesis effort. So how we're processing the index sites. And so we wanted to, and Jeremy had this, so we've been discussing index site processing, and Jeremy raised this question of, well, shouldn't we be thinking about the science questions? So what do we want to ask of the index sites? And those can certainly be related to ecological questions, or they can be related to some combination of methodological. So in other words, how do we process the index sites to ask certain questions about quality control, variation in what we're seeing, those sorts of things. So thinking about that, we wanted to have that as sort of a basis for discussion today. And I wanted to start with some overview of status with respect to the index sites. There's a major push right now to get these inventoried, extracted, and then sequenced. So let me, if I can share my screen, let's see if that works.
Looks great, Kate.
Okay, good. All right, so Karen showed this last time, and then just again to remind everybody, there was some question about, so what are the index sites? And there's been, again, some discussion about what is included, but these are as close as possible to understanding what these official index sites are. So we have these 4 transects starting in the south. It's a combination of lake, stream, estuary, and coastal site in general. So we try to match them up, but there's in some cases, so if we look at Damascotta, we have Damascotta Lake, but then we have sort of 3, I think we're actually calling 3 estuary sites, but one is sort of near coastal. In the Penobscot transect, we have Chimo Pond, Blackman Stream, 2 estuary sites on the Penobscot, and then Hurricane Island as our coastal site. And then down east, we have Gardner Lake, Chase Mill Streams, Bucks Harbor, and we also have missing a point there for Down East Institute as our sort of estuary coastal sites. So those are that's kind of the spatial layout. And this is an extract from Survey 123, And there's the numbers again vary depending on how we've been, what we've said are the index sites. But again, this is those sites which I just mentioned, color coded here. So we have the Damerscott sites, the Downey sites, Penobscot sites, Casco Bay sites. And I've just pulled out from Survey 123 what we have for total filter count. That's all filters. And then broken down by the filter types. So we have, this is a count of the GFF filters that we have, the SUPOR filters, the NITEX filters. There was a couple, just a few cellulose nitrate filters. And then this is just a summary of the field controls and the lab controls, just so you have an idea of what our, again, our sort of our total count is. And this is through December 2022. Okay. So this is essentially what we have to work with, and we'll have a similar, but we're gonna, the idea is now we're gonna continue sampling through October 2023, and that will be the last date. So we'll have another round, March and then May through October of sampling on these index sites. And this is a summary, again, sort of in space and time of the index sites. And we do have some cases where I think earlier, some of the earlier sampling, we maybe missed some sites. I think we maybe have some field sheets that need to be entered into Survey 123. But this starts to give you an idea of our distribution in space and time of the index sites. And I'll just, so again, this is, whoops, back to their sort of general, what is it, jumping ahead, general locations. And then you'll see that there's, with these general locations, there's variations in terms of how they were named. And so this is some of the problems with the naming convention, which was again why we used, we have these index site IDs, and they're associated with the, well, LP stands for Lower Penobscot. These are the USGS hydrologic catchment codes for these different watersheds. So we have Lower Penobscot, LP, so that's Penobscot. MC is the coastal sites for down east. PR is the Presumpscot, so that's the Casco Bay region. And SG is the Sheepscot something, anyway. So we have the Penobscot sites, down east sites, yeah, Casco Bay sites, and then ESG is the Sheepscot. So, and so these are just, well, let me go back. So this was just meant to show what we have in terms of a time series. So if you look at any one year and month, you can see that, so for example, in May 2021, aside from one site, we have essentially a complete time series. Sorry, or maybe June. Yes, June is complete. So you can see where we have, in a sense, complete time series over all the sites. And then this is just flipping it. And so if we now look across a site, and we can then see, so we have this sort of block of time in this center, and that's our typical summer, May through October sampling, where we have fairly consistent sampling starting in 2021 and 2022, and we should have a similar set of sampling patterns for 2023. And for each of those sampling events, this is again a sampling of the data that's collected. So there's information about an observation on water turbidity, observation on wind speed, precipitation. And then for our protocol was measuring basically 2 depths at every index site, a meter from the surface, a meter from the bottom. So typically you'll see for these different, this is Martin's Point in the Casco Bay region. So typically you'll see 2 depths here. We've been measuring fairly consistently water temperature, salinity where it's relevant, and pH and PAR 1 and 2, up looking, down looking, measured turbidity, DO, and chlorophyll. And these have gotten more consistent over time as in 21 and 22 and now in 23, using the SONS consistently to get these environmental measurements. So effectively, we have time series associated with all of the sampling time series. Again, some of them are a little bit more spotty, and not all variables were collected at all sites, but this gives you a little idea of some of the environmental variables that we have. And so what we'll be able to do is, again, sort of also look at, in addition, once we have the sites sequenced, hopefully we can then sort of look at what we see in terms of year-to-year variation in some of these environmental variables. This is just temperature on the Upper Demerara site for 2 years. Didn't get the full year 2022. And then looking at also salinity for the same site, comparison year to year. And so we have some of this data that can start to support some of these possible synthesis questions. So I just wanted to give that as an overview of kind of what the status is of our index sites in terms of the sampling time series and also the environmental time series. So let me stop sharing there and see if there's any questions about that.
And just quick question, Kate, do you have for that environmental data where it's in 2022 stopping in August, do you have a feel if that's just real or just the data is not entered?
Oh, that was just that it hadn't been entered at the time that I made those plots.
Yeah. Oh, yeah.
So we have it through October.
Okay, great. And do you have a feel if some of these, some of the sites where we have gaps here or there, some of those may actually have filters, but again, not be entered, or?
I think so, and that's what we're doing, kind of trying to get
the inventory,
right. Completing the inventory. There could be some that people had field sheets, and so the field sheets were typically done for every sampling event. Some of them may not, for various reasons, didn't get entered into survey 1, 2, 3. So we're doing kind of a final roundup and see if we can collect all the field sheets and get as complete a coverage as possible.
Great.
I have a tangential question, and I'm sorry if this isn't the right place to ask this. In the future, should we continue sampling sites that aren't being analyzed from these 4 transects for index sites? I've got a site at Bailey's Island that I've been sampling since 2020, and just not sure if I should keep sampling there or not.
So have you been doing regular sort of on the same schedule as the index sites, Sydney?
Yeah, I have. Yeah.
Okay.
There must have there's been some miscommunication earlier on that I wasn't aware about. I've also been sampling at the Bigelow dock.
Okay. Yeah, I've seen those, and I guess it doesn't. We've sort of said these are the official index sites, but if we have a regular sort of a similar time series, and I know, say, Julia and Grayson have been collecting some other coastal sites with fairly long time series. So we have some really good coastal coverage with your sites and then Julia's sites. So I guess the question is, it doesn't hurt to continue to sample them. I think that would be fine. What we're maybe running up against is budget constraints on everything that we can process. And this gets to questions too that we want to be able to answer. Do we want to give priority to certain sites? And for those where we have a more complete time series, I think that's that speaks to potentially, if we have an interest in sort of seeing what the, let's say, seasonal and year-to-year variability over sites is, that could be an interesting question, and maybe that's where some of us want to focus. So I would say yes, that would be my answer. And anybody else who wants to jump in, that'd be great.
Because I'm so sorry, I didn't mean to cut someone off. I just don't want to extend my own time or waste project resources if the samples aren't getting analyzed. Or maybe waste is a strong word. Yeah, I do have a time series that coincides with the index sites at both the Bigelow Dock and Bailey's Island. Not sure. Sorry, I've missed that I've been sampling erroneously at both these places.
I think Sydney, and Mike, maybe you can correct me if I'm wrong here, but I think some of that, at least the Bailey's Island is one of Julia's coastal sites. So that's going to this complementary analysis. So it is very valuable that you've been doing that sampling. And Julia has been thinking about whether or not to continue that in a separate conversation from the index site sampling moving forward. And so we will make sure that when that decision is made, that gets communicated very clearly to those of you who've been contributing to that coastal site sampling, which has been much appreciated.
Okay.
Is that helpful, Sydney?
Thanks for that, Christina.
Yeah, it is. That is very helpful. And is okay. That is really helpful.
So for some types of analyses, Julia, yeah, is probably planning to process some of those similarly to the other index sites. So they could be a bigger data set for people to draw on for some types of habitats.
Okay.
Yeah.
I mean, is this a value to your work too, Sydney?
No, this is not the site that I'm using for my work. Sorry to derail the conversation.
No,
I wasn't able to attend the index site meeting the other week.
Thanks, Sydney, for jumping in, and appreciate all your work in terms of doing the sampling.
Yeah, and there's more sites like that again people kind of tacked on at varying points for various questions. So
yeah,
it's not the only
it's the science
one
of team science, right? It's hard to coordinate so many people over such a wide area. So it's totally fine. Thanks.
Anybody else? So maybe I'll turn it over to Jeremy at this point.
Yeah, that sounds great, Kate. Thank you. And thank you for that overview. And Sydney, thank you for your comment. I think we're all thinking about, well, how can we make use out of those great samples Sydney has collected? So what I've done is I've prepared a Google Form survey, an anonymous survey, just to kind of give everyone a chance to chime in their opinion. There's been ongoing discussions among a group of us about the index site samples and trying to make decisions on how to process those. But I'm still really interested in hearing, as many opinions as we can get in that decision-making process to make sure voices are being heard that may not be being heard yet. And then also just kind of the topic for today's discussion about synthesis questions. What are the interesting scientific questions that the index site work is, I think, on purpose been very sort of broad and open-ended, and I think that can be an advantage. We're really interested in analyzing the spatial and temporal variability across these 4 watersheds. What is that variability and so forth. But can we refine those? Can we come up with more questions, refine questions further? And so I just want to pick people's brain who are on this phone call here to see, if there are any burning questions they've been thinking about the index sites, or maybe just to start thinking about those questions, whether they're specific or more broad, sort of, how can we maximize our impact with the index site work? So if people are game, I'll go ahead and send out this Google form. And Kate, how long should we take to fill this out?
I think, let's say no more than 10 minutes.
All right, that sounds great. Yeah, I think giving that sounds like a great amount of time, giving people enough time to think about these questions a little bit, and potentially write down some reasons for their answers, and also whether they have interesting questions to share. So I'll go ahead and send this out.
Jeremy, can I ask, oops, I'm sorry, can I ask a quick question? Will
Yes.
other folks that aren't able to come to the research forum, will they also be able to contribute to this forum? Like, is it?
I see no reason why not. We can share it more broadly. So go ahead and take 10 minutes. So it's 11:26 now. So at like 11:35, let's come back together and go over the results.
Jeremy, I'm getting a permission request.
Okay, let me change
Let me
the
see.
settings on here.
Oh, maybe it's
maybe
the
I'm
big one.
logged in as the wrong person.
It needs you to be logged in as a Maine person.
Yeah, well, I'm
Let
logged in.
me adjust this. So that is Dave and Robin, you're in the same boat?
I think anybody without a Maine email address.
Who else on this call? You could.
Dave Ernst is.
So I sent you an email, Dave and Robin, and I'll get to Dave Ernst. See if that works.
Unfortunately, I'm still getting that. I have the earlier one you sent me. I could I could send that to
Actually, that's a good idea. I have edited that. So I can share that in the chat.
okay. I've got it. Do you want me to share it? I've got it right here.
Don't share that one because I've oops.
Oh, that's a different one. Oh,
Yeah.
You modified it since then. Okay.
Yeah. Just hang on a second here. Okay, I sent it in the chat.
Okay. I can, yeah, so we can just. We're not interactive, but we can still look at the questions and answer them.
We're not going to have your data in the results. I said in 1 more minute to be finished, but I only have 3 responses so far. So let's just see how it goes. If people need a little more time, we'll give a couple extra minutes.
Hey, am I correct that you Maine people can't access it, or sorry, I must have missed something. I can see the PDF, but I cannot I don't have permission to get into the form. Oh, now I do. Never mind. I'm sorry.
Okay.
Until 2 minutes ago, I didn't have permission. Okay, I'll answer now.
No, it was the Bigelow folks that were having trouble.
Oh, I couldn't get in it either.
Huh. Sorry about that. Hi, everyone. Let's give it 2 more minutes. We have enough time to look at these results. All right, let's give it 1 more minute. So if you're not done yet, that's okay. Please wrap up. Doesn't have to be profound. This is sort of just starting thinking about this for some of us. All right, so if you could please send in your responses so that we can go over these. Got 13 so far. The 14, are there more out there? Well, why don't we go ahead, Kate, should we go ahead and go over the results for what we have so far? Okay, let me share screen. Okay, so this first question was about the current plan for the majority of index site samples is to extract 1 GFF filter per water depth. Will this level of replication be sufficient to accurately infer back to the corresponding water sample? And the top answer here is it depends. Although yes is also second, and no is getting some votes, 2 votes for no, 2 don't knows. So So some of the reasons for the answers, it depends on the taxa of interest and their abundance. Studies have shown one filter to be sufficient. Additionally, each filter will have multiple analytical replicates to confirm. There are a lot of variabilities. Replica filters were drawn from a single Niskin. Are technically 1 reps. Yes, not spatial and temporal replicates. That's true. Have not seen any definitive research on this. I think this will be sufficient for common taxa, but might miss rare taxa. Previous data on 16S that we could look at. My sense might be fine for microbes, but not sure about higher microorganisms. All right. So any thoughts to share on the question 1?
I just want to say that there's some studies out there, looking at variation between replicates for different things. So that might be, I mean, we don't have replicates to see where we fit with that, but it might be useful to kind of just put together a meta-analysis or something. If the people are really interested in it.
That's a good point, Aaron. And I was also hoping that perhaps people from their own research in this project have looked at that potentially. If people are collecting, say, triplicate water samples.
Yeah, I've done that in my studies with ports around the world. And yeah, if you want to get species richness, it's all over the place. You need many more than 50 samples, which is not feasible for anybody. But for just general patterns that are consistent, it's like 2 to 3 samples per site. But you should be pretty good. We should be pretty good with 1, if that makes sense to everybody. And then, yeah, species by species, depending on their abundance. Yeah, I think it's really dependent. So people would have to dive down for that into their organism of interest.
I think one of the things that comes into it, factoring it in as well, is it's not just about the individual sample, it's about what they're being compared against. And we've got pretty large spatial and temporal scales that we're hitting. So in that sense, the variation among is probably going to be large relative to the variation within. So it's kind of the ANOVA logic kind of helps us here.
All right, let's go on to question 2. Do you have a preference for which filter types are extracted for DNA from index site samples? The top choice at 40% is C, GFF with subset of those samples, also SUPOR. We also had votes for no preference, as well as SUPOR. In that. So scanning through these responses. GFF passes more volume and thus is more likely to adequately encounter macroorganisms. SUPOR covers bacterial size fraction.
What pore size were the SUPORs? Sorry, I know people know this, but that we used.
0.22.
Point
0.2 or 0.22, I'm not sure.
And did they put a 1 full liter through, or was it just till they clogged?
Generally, it's 500 mils.
500 mils.
500 mils through the SUPOR and 1 liter through the GFF.
Got it. Thanks. Sorry to interrupt.
No. No real preference, but CN has been working well for me, and others use that filter. I think filters do about the same in terms of eDNA collection, but it'd be good to have an empirical data showing this. SUPORs are a little easier to work with in the lab. So I'd understand if the extraction techs wanted to do those. Okay. 3, do you have a preference for which genes are sequenced for metabarcoding of index site samples? Pick the best answer. And 50% said F, which is 16S, 18S, 12S, and CO1. Second option was no preference. And 20% said 18S, 12S, and CO1. Okay. Okay, well, that's interesting. What is the reason for your answer? It seems to match best with group interest. We want to characterize total biodiversity, hit them all. Still new and beginning the process, and we'll need to try more genes for sequencing to see which I prefer. Whatever works best for the group. These are the broadest set of barcodes for covering plankton to whales. 16S would be nice. But Davey has suggested that water samples aren't the best for microbes that he and Jeremy are interested in. Universal bacteria and universal eukaryotes covers everything, but is a jack of all trades. I have a microbial bias. Should give us the broadest taxonomic coverage. Don't have enough information. More diversity might be better. Okay, this is great. 4, what is most important to you in deciding priorities for which index site samples are sequenced? Pick the best answer. And D had the most votes, more complete spatial and temporal coverage with less taxonomic coverage. And then B was second, temporal coverage, and then no opinion, with 2 people saying more complete taxonomic coverage with less spatial and temporal coverage. So the top answer here, more complete spatial and temporal coverage with less taxonomic coverage. So reasons, we have the data to do both. The original questions in Maine eDNA proposal relate to eDNA diversity patterns across space and time. Spatial and temporal questions are much more interesting. Change in time may be most interesting. More complete spatial and temporal coverage gives us better power to answer ecological questions and maybe management relevant. Impacts on fish migration. Preference for more diversity, but I can see the value of more spatial and temporal coverage. So we can focus on more bigger picture statewide questions with higher resolution at certain sites, perhaps those that have better spatial temporal resolution. All right, great.
I really
Question.
like that. I really like
Yeah.
that last comment on there is a really good, justification too, because, with eDNA, one of the things that we sort of put out early on is that, we could address something bigger than would be easy, able to do with other approaches. And that's often one of the pieces that, we kind of struggle to get up to, that whole state coast scale. So I think that's a neat response. Kind of gets us to that macro system scale.
Thanks for pointing that out, Mike. Other comments to share here?
I think one quick comment that this question seems like the one where there's actually the most consensus of the questions that you've asked thus far. That's a sort of general sense I'm getting, so maybe I'm not quite right there, but that's interesting and maybe helpful in thinking about how we address some of the inconsistencies within our group in the above questions, if most of us are in agreement about large spatial and temporal coverage as being valuable for this part of the project.
Yeah, I would just add, this is probably the most important question, right, in terms of synthesis. Most challenging to answer too. But it's nice to see that there's a more consensus than there might have been.
No, this is helpful to see just in terms of how we're going to be making decisions. Question 5, what are the most interesting research questions for you personally? Invasive species distributions. How eDNA can be used to identify what species are within an environment, related to dispersal, how regions differ, cross-habitat comparisons, are lakes more consistent over time, but less consistent over space than coastal? What species are present at different sites, how communities shift, community structure change along the transects, ecological networks, influences of the pandemic, seasonal anoxia in freshwater lakes, abiotic and biotic factors contributing to white shark species range shifts. Do prokaryotic, eukaryotic, invert, and fish patterns correlate with each other over other of time and space? What microbes are associated with macroalgal habitats? Any thoughts or comments to share on question 5? I think it's great just to see what people are thinking regarding this question.
Yeah, so I couldn't write in the chat in the form, but I think the two things that I had were both covered in there, which is sort of linkages across taxa, as well as just understanding dynamics of change across estuaries between and coasts between dominant taxa. And I think that's a question where, you know, if you're going to do that broad comparison, it's going to probably have to be on the more dominant taxa, versus if you want to look for rare things, that's probably another set of questions.
There seems to be a fair consistency in these, or there's similar ideas expressed in these, which is also good to see.
Yeah, so it's nice to see that. I didn't have anything original to add.
Others? Well, we have 5 minutes left here, so let's go to this last question. Thinking about the broader group, what are the most important research questions regarding the index sites? The ability of eDNA to detect changes over space, time scales, feasibility of environmental monitoring, collaborative effort, take in mind sample bias, many different hands were collecting and filtering these samples, scaling, species presence and how their abundance changes, comparing taxonomy phenology between transects, baseline for future climate change studies. How well do eDNA results reflect seasonal and spatial patterns and aquatic diversity that we expect or know from more traditional sampling approaches? Anthropogenic stressors, effects on species and community diversity. Pretty open to anything. Statewide fish diversity could be great. Where and when do we see species of interest entering, leaving areas within the Gulf of Maine? That's an interesting question. How do trends in various communities co-vary with major trends in environmental correlates? This is great. Great to see these questions. Any thoughts coming to mind about question 6?
Yeah, I just jump in again. Pretty well covered with my thoughts. The one thing that I had was also, I think, is that these could be, how useful are these sites as benchmarks for other people's research questions, which I think is also important as either control or a benchmark. But it's great. Yeah, no, I sort of, yeah, I had the team science thing, which I think several people brought up, as well as just the whole question of how much sort of the broader scale inferences you could make with this kind of data set.
So when you say team science, Dave, you just mean the general collaboration among the group?
Yeah.
Yep.
And I think that's covered. Yeah, there's the collaborative effort to establish Maine as the eDNA coast. I guess that would be another way of phrasing it.
Jeremy, there might be another question in this, or not a question, but more of thinking about how we approach this research and what kind of advice or thoughts we could share with the community if someone was going to set a study up like this again. What would people suggest? How would you go about it? What would those efforts look like? So that could be really, really helpful from everything of how do you do reliability, from filters to publication.
So what have we learned? What might we do differently? What advice would we give? Those types of questions.
Yeah, it could be really helpful.
Yeah, I would say lessons learned from such a big complex project. Yeah. Good thought, John.
And Jen, I also liked your suggestion about sending this survey out to the broader group. I think I would like to do that. And I also want to figure out how to turn off the UMaine only settings.
I think it would be beneficial, Jeremy, for everybody to have a potential voice on this, especially because we're doing index sampling right now, aren't we? So some folks just don't have a chance to log in, or they're teaching, and they might have really good thoughts about this. I'm sure they do.
Yeah, that would be great to get wider input on this if we can.
Yeah, I agree. I think we should put a pretty strict time limit like this week on it, but because we
By
are
the end of the week,
forward
perhaps.
pretty quickly.
Yeah.
But I mean, the questions here are great. I think there's a lot of food for thought in terms of, and those responses are, yeah, I think that's going to be very useful.
Or what if you make it to next Monday? That would be a week. I only suggest that in case anybody is in the middle of comps or they're just not able to take it.
I can do that.
I know anybody's taking comps this week. One possibility too that I'll just put out here for people Again, our next research forum, we have Jerry Ziegers up, but we do have one in April that's open. If we thought coming out of an exercise like this, there was an idea to reflect back and come in and ask, now, how do we do some of these? Like, how do we make some, happen in person? That could be another use for that time, especially after we get some of this consensus, we could think about what an analysis design might look like to address some of these. Just put it on the table. But I'm also open to other suggestions if people have ideas for that session.
I think that's a good suggestion, Mike. Thank you. Other parting thoughts before we get off the call? Kate or Dave?
No, this was great. Thanks everybody for coming and taking the time to put some really great thoughts down. I really appreciate that.
Yeah, I think this has been useful, and it's also just useful to see all the data start put together. Thanks, Kate, for organizing that. And can we share, is that being, maybe it would be good to send that out as well with the survey?
Yeah, sure.
Those slides you had?
Yeah, maybe we can put them up, either send them out or put them up on a site where everybody can access them, including the people not at the University of Maine, hopefully.
Yeah. All right, everyone. Thanks for joining today.
Thank you all.